have a very good laboratory where I work, and I'm going to try to give you a little bit of a snapshot of what we do. It's going to be a very little snapshot because we do a lot and we only have a little time and it's late, so and everybody had a long day. So I'll try to keep it as brief and as interesting as possible. Now, as you have probably seen already, I'm old. Okay? I still remember that when the phone was installed in our apartment where me, my parents, my brothers lived. And it was a rotary phone. It was super exciting because we didn't have to go to the city market square to call anybody or some rich neighbor who had a phone. So it was absolutely cool. At the time, I would have never imagined taking my phone to the beach to take a picture or anything like that because it was just not imaginable. Nowadays, we use the phone for so much more. The phone has totally changed. And the question is, how do we get from here to there? And I'd like to apply that to how we're using precision medicine and how we're trying to get from here to there, and what is needed, and what are we doing in our laboratory to get from here to there. Now, one of the signs that I'm getting old is when my normal life memories are being painted in sepia colors on the internet. So, okay, precision medicine. Targeted therapy. We mainly focus on cancer research in our laboratory. I worked in Alzheimer's disease before, but right now we do a lot of cancer research. And when I talk about targeted therapy, I'm trying to contrast that with general chemotherapy. So what most of you probably know is when chemotherapy, what we're trying to do is basically kill any cell that divides rapidly. And we're basically giving you poison and hoping that the cancer dies before. But many times it works the other way around. It's just, that's the nature of it, that's the way it works. We're trying to push as far as possible, almost to the brink of death, and hope that the cancer dies first. Now, targeted therapy tries to go and find something that only the cancer will react to. That's difficult because the cancer is basically a slightly changed version of you. It's not an infection, it's not a foreign organism or particle, so it's not that easy to find something that is particularly specific to cancer. But there are certain approaches, and there is hormone therapy where cancers are responsive to hormone treatment, or that they are dependent on a certain amount of level of hormones in the body, so you can give something against that. Or immunotherapy, where we're trying to charge the immune system to actually go after the cancer cells, because they're really, really creative and very good at evading your innate immune system, trying to get away from that. And then angiogenesis inhibitors, monoclonal antibodies, and so forth. But the only thing I'll try to talk about today is signal transduction inhibitors. Now, in our laboratory, we work mostly with proteins. I know that when you talk about personalized medicine, precision medicine, at least in the media, it is always geared toward gene therapy, gene analysis. But in our laboratory, we work mostly with proteins because we believe that proteins are the actual action items in the cell. The DNA is basically where it's written how to make things, but what really happens is at the protein level. The changes, secondary modifications of proteins, and that's where the action really happens. And that's where most of the drugs actually happen. They usually don't, at least at this point in time, we don't change genes inside patients right now. We mostly target proteins. So that's what we're trying to get at. Now, signal transduction is something I absolutely hated in school and in university. I promised myself as soon as I leave university, I'm going to get as far away as possible from it and never touch any book or anything like this again. And now I do it morning to night every single day. Not because I have to, because it's absolutely cool. But that is only cool once you actually understand that it's cool and what is really interesting about it. And that's the hard part, I believe. And mainly is because we are not good at comprehending very complex information as human beings. Or maybe I should talk about myself. I am not very good at comprehending very complex networks. I like to think of things linear, okay? So when we talk about signal transduction, I like to think of pathways. There is a ligand, you have these little red dots or green dots, something enters the cell, the yellow lines are supposed to be a membrane, and there is a protein at the membrane level, it detects that there's a hormone or another small molecule is coming by, binding that, then it's transmitting that signal to another protein, another protein, another protein, another protein, and then in the end we have a change. Either the DNA or the cell reacts in a different way, or it's starting to proliferate or change in any way. Now, I thought about it, but decided against showing any chemical structures. I thought it's probably too late for anybody to want to know that chemical structures. So I'm just going to, we'll just call this simple, okay? The protein gets phosphorylated because I don't know everybody's background. So it means the protein gets a phosphate group attached to it. What that does is that the protein now changes its conformation. There are pockets that open up, and that means the protein is now, let's call it active. So it can now do what it needs to do. It can either bind other proteins, it can change other proteins, it turn into an enzyme itself. And an enzyme, in this case like a kinase, is the one that helps proteins get phosphorylated, and a phosphatase is an enzyme that helps get them dephosphorylated again. Because you always have to have the two, right? You can't just go into a room, turn on all the lights, and never turn them back on. That would not be a normal system to work with. So, with all that out of the way, so this is when we talk about signal transduction. One protein phosphorylating, another protein phosphorylating, another protein, so they're all getting turn on, turn on, turn on. Now, as scientists, we like to take things apart. We want to take at least little bit and understand it. So this is like a slide that I use when I talk about insulin pathway. Okay, so here for the students, I take it apart. I look at all the different proteins, who does what class of proteins are they, who's binding who, and then what do they do with each other, and how do I get the signal from all the way up here to the insulin receptor down to the DNA level. Okay? And insulin is really cool to talk about because although we always think about it in terms of pancreas and sugar, in reality insulin is everywhere. It's in your brain. People have done studies where they sniff insulin, it helps the memory, there's all kinds of things that happen to insulin. But this is taking signaling apart, and that's where I usually get lost, and that's when I hated it in school. But if you take a step back, what signaling actually looks like, it's not pathways, it's more like this. Okay? Now this is a single pathway. What I showed you before, just let's say A, B, C, D, E, that's just for one receptor. This is the epidermal growth factor receptor pathway. So when I say pathway, that's what I mean. Now that's where things get really interesting, because now I can start to understand how a cell can make a decision based on information. Because when I look at this looks a lot like a computer, okay? Like a motherboard on a computer. And that's basically what we're trying to do when we look at signaling. We're trying to understand what a cell was thinking. What was the decision process in that cell to come about with this conclusion. And another way for me to think about it is like a train map. Okay, so this is me young. I know it's hard to recognize. That was the love of my life back then, and I'm still married to her still. I was living in the south of Berlin. She was living in the north of Berlin. It was about an hour and a half by train. What I had to do is get on a train here, right up to here, take another train, go up there, take a bus, and then see her. Now, out of all the 4 million people living in Berlin, the only person I wanted to see most of the time was her. It all depended on timing and location if I saw her or some other random person. If I get on a train at a certain point in time and get off that train at a certain point in time, I'm at this station. Now, depending on how long I wait for the next train, I'm going to go over here and see random person X, or I'm going to go over here and see my girlfriend. And so timing and location are extremely important for signaling. Where in the cell does it happen? What happens before? What happens after? And that's how you can start to understand how the same proteins, how the same nodes in those pathways can suddenly have totally different outcomes. That was something I was really struggling with at the time, because you see the same proteins come up and up and up again in all kinds of pathways, and if the cell's supposed to proliferate or differentiate, you have the same proteins involved. And I couldn't understand, because how can you have a different input, the same proteins involved in a different output? How is that possible? But it's all about timing and location, how they interact with each other. Okay, and then in the lab, I can produce maps like this. Okay, so for example, this is from, I believe this was from a brain cancer patient. So I get a tissue sample, and I can analyze it, and make a map. This is supposed to be a map of the cell. We have the nucleus right here, and then different proteins drawn into that map to show, okay, this patient had certain pathways upregulated or downregulated. So red would be hot. This is a very active protein. Green would be very low activity. And the funny thing is, or interesting thing is, even if people have the same disease, in the cell it looks very different. Everybody is very different from each other. And we still don't exactly understand how that's possible. But on a cellular level, we're all very different. So when we want to do targeted medicine, we'd like to have this information on everybody and then decide, okay, we're going to give you inhibitor A or we're going to give you inhibitor B. Now something that I usually do in class is when you start to think about what does an inhibitor actually do. The cell is extremely smart, and the inhibitors don't always work the way you want to, because imagine you take out one little protein here, what's going to happen? We have no idea what's going to happen because there are 50 different pathways around it. And so it is it's pretty complicated, but this is where we want to be. We would like to have this information, but at the current level in the clinic, we don't. So how do we get from a single patient to having an actual map of their cells? What are the cells thinking right now? Why do they come to the conclusions and decisions they are? And I like to use the same analogy. So how did we get from here to there? And I think the key is, if you take the phone apart, there are many different developments that had to come together for this to work. Nobody, when I was a child, sat down and said, man, I wish I could take my phone to the beach and take a picture. It just wasn't there. But once he had a lot of development, somebody developed a touch screen. Another one made sure that computer components get really, really, really tiny. And then you had Wi-Fi development. All that came together at the right time, and now we have the smartphones. So I'm just going to give you a quick snapshot of what we're trying to put together in terms of technology to make precision medicine in the normal clinic possible. Okay, so when is the last time you saw this at your doctor's office? So I have this in the lab. It's a liquid nitrogen tank. We keep cells alive in there, keep them stored. You can keep proteins in there forever. They don't change. But you need liquid nitrogen. You need people that know how to handle this. And you do not have this sitting around in your community clinic. But preservation is extremely important. Okay. So this is a picture of two mummies. I know there's a little age difference how long they were preserved. But what I'm trying to say is that it's extremely important how your biopsy, how the tissue that a doctor collects is preserved. This is a study that a colleague of mine did. So what she did is she took a uterus sample from a patient, okay, out of the OR. There's a time gap here of 30 minutes because you had to get it out of the OR, the pathologist had to sign off on it and so forth. But then she left it sit at room temperature, just right there, and took samples every 10 minutes. And what you see here are phosphoproteins and cleaved caspases, so all secondary modifications of proteins over time as the tissue is sitting. Okay? You see dramatic changes. And what's interesting and what nobody really expected is those changes don't just go all down. So we're not just talking about disintegration, degradation, everything just falls apart. No, you see a lot of live action. And that's something that we usually don't think about. But if I cut off my finger, put it right here, apart from it being very disgusting, tomorrow a lot of the cells would be alive. They would still be alive and sitting there. They don't just die that quickly. They just sit there. They suffocate. They're sitting in their own waste products. They don't get any blood supply. None of the normal signaling is happening. They're struggling to survive, and they do all kinds of things that have nothing to do with my finger when it was still attached to my mouth. And the same thing happens to a biopsy or tissue sample taken from a patient in the OR. It's taken out of the body, and then it struggles to survive. When I, as a researcher, or whoever analyzes it 2 hours later, get that sample, what I'm measuring, the path map that I do, may have nothing to do with it being cancer or whatever disease. That's only thing, the biggest thing that I see is going to be the struggle for survival of that piece of tissue. That's a big problem because right now it's very hard to preserve tissue so we can do everything at once. In the clinic, what is normally done, you have a piece of tissue, it's being preserved by taking all the water out, and instead of the water you put paraffin in. So we keep tissue in a block of wax, basically. It's extremely stable. You can keep tissue for decades like this. Morphology looks beautiful. You can cut sections, it looks just like if it were alive. It's absolutely gorgeous. But the labile biomolecules, most of them are gone or changed because fixation takes a long time. You need to cross-link proteins, and it just takes so much time that a lot of the changes already happen. Now, on the other hand, we can quickly freeze tissue, drop it in liquid nitrogen, put it in -80 freeze or dry ice, and that'll preserve all the labile biomolecules I'm interested in. But anybody who's frozen strawberries before and then thawed them again knows what it's going to look like. You can recognize a strawberry, but the morphology will be awful. And a pathologist who needs to make a clinical diagnosis can't do anything with it because it just looks horrible. They do frozen sections, but it's very hard to work with, and nobody really likes it. So the current solution is we pick 2 biopsies. We take one that we freeze, and we put one in paraffin. The problem with that is now I have 2 different samples, and the tumor on this side and the tumor on that side may be totally different from each other. So it's very good. Plus, who wants to have 2 pieces of brain data? So what we did is, okay, we need to solve this problem. We need to come up with something that works at room temperature that any random clinician can have in their clinical office, and that's not a problem to work with. So we came up with Theralin. It's patented. It's available commercially. And what it does, it preserves all the labile biomolecules just as if you had frozen it. And it preserves the morphology just as if you had fixed it with formalin and put it in paraffin. It took me like this side of the head of gray hairs to develop it, but at least it works. So here's a picture of this mouse liver, snap frozen in liquid nitrogen at time 0, and another piece that we fixed with Theralin and then put the paraffin block, kept it at room temperature, and we analyzed phosphoproteins and saw do they look like they looked at the time we froze it or not. And overall, it's very comparable. So we don't see a lot of changes, even if we keep it at room temperature. And at the same time, if we look at morphology, so BHP is what we called it at the time. This is what I mean to tell. And we compare this to the standard of practice, which is formalin. We can see very beautiful morphology. You can see different layers of the brain. Here we took tests of one of my coworkers. Her cat was neutered. So we took the tests and put them into the fixer, and we see a lot of chromatin detail which we don't even see normally. So it works really beautifully for that. Okay. Now that we have everything preserved, everything is fine. Now we need to look at it. My youngest son, he's extremely picky here. Anything with colors off limits. And it's a huge fight every single night. I'm always looking forward to it when I go home. But being picky is not always bad. Let's assume you have a piece of tissue. Okay? This is colon cancer. And I hope it's pretty obvious. There is a tumor right here, and then there are a lot of other cells. Okay? If I want to develop a targeted therapy or make a decision if I want to give you this therapy or that therapy, what I really want to know is what do the cancer cells look like? What are they thinking right now? What drives them to behave the way they do? I don't want to know about these cells. They just mess up the picture. If I really want to find out the orange gummy bears, what do they taste like? I can't just grind up all the gummy bears and then kind of get a feeling for it. I got to look at what I really want to look at. The problem is, this looks nice and big on the screen, but this in reality is probably a little less than a millimeter thick wide. Okay, so you can't see it. I can't just scrape this part off. And So what do? This is a method that was developed by my boss, and it's actually in the Smithsonian right now, the machine, because it's pretty ingenious. What he decided to do is take a laser and shoot the laser through a little plastic piece that has a polymer at the bottom of it. Okay, so I take the little plastic cup, put it on top of my tissue, and shoot the laser through. The laser is going to melt the polymer. You see this here as a little ring. And where it's melted, the polymer is going to connect and stick to the tissue that's underneath. And when I lift off the cap, I'm just going to remove all the cells that are stuck to the polymer, and then I can lyse them. And all the rest that I didn't care about is still down there on my slide. So, at least the older machines that we had, it was a lot like computer playing. Anybody young was happy to do it because it's exactly what you sit there. You have a screen, you see a tissue, you have a joystick, you shoot the laser, and the other hand moves the tissue, and then you just go collect as many cells as you can. Now it's all modern, you just click and draw, and the computer does it for you. But we had some people get really seasick from doing this, staring at it a long time and just moving the whole thing around all the time. It was not everybody's job. But what it looks like in reality is this. I hope you can see it. This is a mouse brain, okay? And the mouse has brain cancer. Those cells are all cancer cells, and this over here is normal brain. Okay? So this is what it looked like before, and this is what it looked like after microdissection. Here are all the cells that are attached to the polymer, and this is what a cap looks like. And then I add some lysis buffer. Just lyse all the cells, break them open, and look at the proteins and what they look like inside. Now, does it matter? That's the big question, and that's even was a big debate among scientists. Does it really matter? Is this very important? So I did a study on brain cancer samples, 39 patients, and I compared just lysing everything versus just microdissecting out the cancer samples. And just to summarize the study in a single slide, basically what I found, even if I take pieces of tissue where 90% of everything is tumor, there's just a few cells that are non-tumor cells mixed in. For some proteins, these are phosphoproteins, I get, for example, here 40% misclassification. So if you were a patient that needed to get an angiogenesis inhibitor, something that targets VEGF receptor R, and I look at your sample when I don't microdissect, and I look at your sample after I microdissect. 40% of the time, I would misclassify you as either getting the drug and you shouldn't, or not getting the drug when you shouldn't. So there's a huge difference in microdissecting or not microdissecting. And one thing we don't even look at right now, we're just starting to, is something that students that you teach will have no problem with, but somebody as old as I am is just slowly getting into it, is that everything is connected with each other. We've been looking at right now at cell signaling inside a cell. So how is the cell perform? What is it thinking to do? But there's another layer of complexity because all the cells are not just deaf and dumb. They just sort of sit there and don't see anybody. That's the way we envisioned them maybe before. But every cell is talking to all the other cells around it. And there's a lot of communication and interaction taking place that we can't see, we can't measure. We don't know who's talking to who. We can't see this kind of interaction. Very limited cases. So that's something that the future generation will have to take a look at. Okay, for my last little part, I just want to highlight one thing though. We talk about targeted therapy. You would think that targeted therapy is very specific, as opposed to cancer where you have all these chemotherapy, where you have all these side effects. Now, targeted therapy should be just right on the target, and you get exactly what you hope for. But think about it this way, okay? Everything that a cancer cell has, most of it, every other cell has too. The cell signaling pathway that I showed you is not a specific cancer way of thinking, but it's just using normal communication, a little out of whack, that every other cell in your body does too. So if I give an inhibitor to a certain part or or a certain node in that cell seeming pathway, that's going to affect all the other cells as well. Hopefully not as much as the cancer cell, but it will have some effects. So let me give you an example. This is talking not about cancer, but just simple smooth muscle relaxation, okay? It's actually a very complicated process. Somebody got the Nobel Prize for this because it involves three different cell types, plus a lot of cell signaling inside. We won't go into the detail, but it's very important for you. Hypertension is a common problem for many of us. What you need, neurons, you need endothelial cells, you need muscle cells, and there's a lot of crosstalk between the cells to get your blood vessels or your smooth muscles to regulate how thick they are to open up, to relax. Now, one key molecule in this is CGMP. So I can do anything more than twice a day. So I'll not go into the detail, but just remember the amount of this determines how much your muscles have relaxed. And the key enzyme that regulates this is phosphodiesterase. So the thinking was, okay, if I have patients that have angina, somebody has a heart attack, what do? I need to make sure that their vessels open up immediately, they get blood. So what do I need to do chemically? I'm going to target phosphodiesterase. I'm going to give a targeted phosphodiesterase inhibitor to make sure that we have a lot of GTP, very little CGMP, and the vessels just stay open. Okay? And so they tested a lot of drugs, and they came up with the UK92480, which is a phosphodiesterase inhibitor. It worked. They gave it to patients, did a clinical trial, thought, okay, let's see what the toxic side effects were. And the side effects were there, but they were not what anybody expected, because that's the drug that came out. And it does have certain side effects that make many people happy and the company very rich. And it does what it's supposed to do. It inhibits phosphodiesterase. And it keeps vessels open. Okay? But it means targeted therapy doesn't mean there will not be any side effects. It just means that hopefully they're not going to be as bad as we see with chemotherapy. Okay, that's the end. That's just a quick little snapshot of what we do in the lab, among all the other things. And I have also talked in high schools and presented what we do there, and I'd be happy to do that with anybody if you'd like to. I think it is very important to get young people excited about this in a way that they can dream. At least I always have these fantasies and think about how cells are thinking and what are they doing, what can I do to make that happen. And I think if you can get kids excited on that level, then it makes a big difference. Because we all need the knowledge itself, the balls, so to speak, and we all need the creativity to make it juggle. Just the balls is super boring, just the juggling looks stupid without the balls. But if you put that together, then you can make a lot of things.